Thank you very much for the kind introduction. It's actually great to be back here again this year to represent ProBio. And today I'm going to speak about the overview and outlook of conjugation technologies in AXC. So I'll first start with introducing the concept of AXC and various conjugation technologies, and followed by an overview of ProBio CMC platform capabilities to enable the AXC manufacturing. And finally, I'll provide some future perspectives in this rapidly evolving field. The AXC concept, or somebody call it antibody plus, is a rapidly evolving term over the past decade. And in this term, so antibody plays the role of specific targeting, and the X plays the role of high efficiency function. And basically, in this X, this could represent a variety of different modalities. For example, if this X could be a potent small molecule to make the conventional ADC, it could be the radioisotope to make the antibody radioisotope conjugates. It could also represent nucleic acids to make, such as ASO or siRNA, to make the antibody oligonucleotide conjugates. It can be the peptide, so to make such as GLP-1 drugs to make the antibody peptide conjugates. And finally, it could represent complex systems such as immune cells and lipid nanoparticles to construct CAR-T, CAR-NK, and targeted LNP therapeutics. So the main purpose of AXC design is to achieve targeted delivery of payload, expand therapeutic window, and reduce the toxicity associated with traditional chemotherapy. So over the past few decades, there has been a lot of evolution for AXC design. So starting with early years with the first generation utilizing animal-originated antibody with high toxicity. And over the years, there have been a lot of improvement in the linker payload as well as the antibody. And we are now at the third generation of AXC, which mostly utilize fully humanized antibody with more hydrophilic linker, more potent drugs, as well as a uniform distributed DAR, utilizing site-specific conjugation techniques. And in AXC design, there are three key components, namely the antibody, the linker, and payload. So for the antibody, it should have a desirable affinity with low immunogenicity. And for the payload, they must deliver intended therapeutic effects without excessive toxicity. And the linker connects the antibody to the drug and should be released at specific conditions at the target tumor or tissue environment. So last but not least, the conjugation technologies basically determines how these key components are being connected and will be the focus of discussion for today's talk. So here is an overview of various enabling conjugation technologies for AXC. So each of them has their own pros and cons. And for example, for the conventional lysin and cysteine-based conjugation, utilizing random conjugation, the key advantage is their simplicity in operation, as well as have the most FDA-approved and clinical tested ADCs in the track record. However, on the downside, these random conjugations will result in a variety of DAR distributions and generate very heterogeneous products and product quality. On the other hand, there have been emergent site-specific conjugation technologies that enable more precise control of the DAR. For example, through the engineered cysteines, cysteine rebridging, on natural amino acids through enzymatic conjugation and glycol-based conjugation. So the key advantage of all these site-specific conjugation techniques are more defined DAR or even uniform DAR and very high conversion rate, a more defined conjugation site, and homogeneous product and product quality. On the downside, there are a couple of challenges for each of these methods. For example, a lot of them will require genetic engineering in the target antibody, so that might impact the stability or immunogenicity of the target antibody. And a lot of process involves enzymes in the reaction, so that will bring in some complexity and increase the cost of the overall manufacturing process. So in the next couple of slides, I'm going to do a little bit deep dive on each of these conjugation technologies. So starting with lysine-cysteine-based random conjugation, basically utilizing the native residues in the antibody. So for the lysine-based conjugation, through amine coupling of the lysine site, since there are about 40 to 80 native residue lysine sites available in the antibody that is able to be conjugated, the resulting products will be highly heterogeneous. On the other hand, for cysteine-based conjugation, it basically utilizes the 4 pairs of interchain disulfide bonds in the antibody. So after the reduction of the disulfide bonds and expose the thiol groups, it could be directly covalently bonded to maleimide groups through microaddition reaction. In this way, the DAR value could be a little bit better controlled from DAR 2, 4, 6, and 8. However, the distribution of the DAR cannot really be precisely controlled given the nature of the random conjugation reaction. So utilizing similar cysteine-based chemistry, conjugation chemistry, we can introduce engineered cysteines at specific or desirable positions in the antibody to realize site-specific conjugations. The most well-known platform for cysteine engineering is the ThioMab platform, which was developed by Genentech almost 2 decades ago. So there are 3 key steps involved in this conjugation process. The first step is a reduction step, basically utilizing a very strong reductant to disrupt all the interchain disulfide bonds, as well as uncap the engineered cysteine sites. The second step after the buffer exchange is the oxidation step, basically is to introduce a mild oxidant to reform all the interchain disulfide bonds in the antibody. And the last step is the conjugation of the linker payload to the engineered cysteine site. So in this way, we can achieve precisely controlled DAR value and homogeneous conjugation sites. And at the same time, the stability of the antibody is improved since all the interchain disulfide bonds is being retained in this way. For the unnatural amino acids conjugation platform, basically the idea is achieved through expanding the genetic coding to allow the expression of unnatural amino acids in the antibody, which contains specific reactive groups such as ketones, alkanes, or azide. After the introduction of these unnatural amino acid residues, a direct coupling reaction with linker payload could be done through bioorthogonal reactions. So the most well-known platform in this area is Sutron Biopharma's platform, where they has established their cell-free expression system, producing the antibodies that contain the unnatural amino acids. Move on to the peptide-based technologies. This is also highly specific, and then basically this can be realized by introducing a specific recognition sites sequence in the antibody, and which could be specifically recognized by a particular enzyme. And on the other hand, also a second catalytic site should be introduced in the linker payloads end. And once both criteria are met, these enzymes could catalyze this site-specific conjugation with very high activity and specificity. And there are a wide range of different enzymes that could catalyze these reactions, with some examples listed here, such as Saltase A, MTG, FDase, and FGE. And lastly, there have been a lot of cases utilizing glycosyl-transferase technologies for site-specific conjugations. So the basic idea is to leverage the natural glycan residue structures in the FC fragment of the IgG, and utilizing these enzymes to introduce some engineered sugar residues that contains the chemical reactive groups such as azide, and subsequently through click chemistry to directly link the linker payload to these specific sites. There have been a couple of different platforms utilizing glycotransferase-based technologies, with the most advanced one from Synaffix, which is called GlycoConnect. And there have been a couple of ADC drugs currently in the late phase clinical studies. After an overview of this site-specific technology space, let's take a closer look at our platform capabilities to enable some of this AXC process. So starting with overview of the conjugation process flow, it involves first the production of naked antibody, or we can call it DS intermediate, which is not much different from conventional MABs or bispecific or other antibody format production. And second part is pretty much the preparation of the linker payload, followed by the conjugation production. And generally, with respect to the conjugation process, there could be multiple steps involved, such as the reduction, buffer exchange, oxidation, buffer exchange as needed, conjugation, post-purification, buffer exchange, and formulation step. So I'll just take two platforms as examples. First, the cysteine-based conjugation platform. And as I said, so this approach basically leverage the different reduction potentials in the 4 pairs of disulfide bonds in the IgG. So for example, for IgG1, typically the disulfide bonds between the light chain and heavy chain is a little bit easier to be reduced than the hinge regions between the two heavy chains. And we can fine-tune the reduction condition to get the average DAR as targeted. So there are a couple of key considerations in the cysteine-based conjugation process, starting first with the selection of reductant. So normally we'll prefer to use a strong reductant, and compare with DTT, the TSAP is more stable with a wide range under different pH conditions and the environment, therefore, and end up with higher reaction efficiency and does not react with a coupling reagent, and therefore is selected as a platform reductant. And also, can we control the overall reduction through reaction time? Not really the case. From this data, we can see at different reductant to MAB ratio and different reaction conditions, all the reactions turns out to be very fast and reach equilibrium within 1 to 2 hours. And considering the manufacturing operation time, it is really difficult to control the DAR value, average DAR value, by controlling the reaction time. And it's actually recommended and suggested to control the overall DAR value through the reduction ratio. And for example, theoretically, if we have 1 mole of TSAP versus MAB ratio, it can break down 1 pair of disulfide bond and result in an average DAR value of 2. And this should be linearly scale up as we increase the ratio of the reductant to the MAB. And which through the experimental results utilizing the model IgG1 molecule, the experimental data agrees pretty well with theoretical prediction, where we can easily achieve the average DAR 2, 4, 6, and 8. However, the DAR distribution cannot really be controlled, and most of this just indicate average DAR and result in the distribution of different DAR values. And taking ThioMap as a second examples, there are a couple of key lessons learned from our development. And first is the point mutations in the IgG plays a key role determining the stability of the mutated antibody. For example, in this particular case, we introduced point mutation and cysteine, engineered cysteine sites at 3 different spots in the IgG, and turns out to have a very significant impact on the purity of the IgG molecule. And just a recap for the ThioMap, there are 3 key steps: the reduction, fully reduction, reoxidation, and conjugation. And actually, the reoxidation is a key step through all this process to make sure that we can fully close all the free interchain cell bonds and allows the conjugation happen specifically at the engineered cysteine site. So it turns out to be the molar ratio of the oxidant to the antibody is the most significant factor determining DAR, basically to make sure as we increase this oxidant ratio, we are essentially making sure that all the free interchain cysteine is being closed. At the same time, the oxidant ratio and also the mutation sites on the antibody, as well as many other factors will impact, such as the antibody concentration, etc., will impact the stability and purity of the conjugated products. Therefore, all these factors need to be carefully balanced and optimized in order to generate a fully optimized conjugation process. And here is an example of optimized process for a model IgG molecule for thiolmab conjugation. As you can see, after optimization, basically all the interchain disulfide bonds should be well oxidized with very minimal DAR value generated from these free thiol groups from the interchain disulfide bond. And essentially, all the conjugation should essentially happen at the engineered thiol group. And the resulted DAR is highly uniform at the target DAR2. And finally, I'd like to speak about a couple of key trends and perspectives for the future of AXC field. First, one of the key observations we have, as we can see over the past decade, especially from the recent years, we can see that ADC or AXC are rapidly evolving from random conjugation to site-specific conjugations for improved homogeneity, stability, and manufacturability. In particular, starting about 2-3 years ago, essentially all of the ADCs currently newly entering the pipeline utilize site-specific conjugations rather than the conventional random conjugations. And if we do a deep dive into the site-specific technologies being utilized, we can realize that the majority of them use enzymatic-based conjugation or glycol-based site-specific conjugations. So starting last year, we also established our R&D platforms to support this emerging enzymatic site-specific conjugation applications. For example, for these particular case studies, we utilize MTG for site-specific conjugation. And basically, this recognize the Q295 spot in the IgG. And also at the same time, we introduce the N297A mutation to remove the glycosylation and enhance the coupling efficiency at the desirable site. And after the introduction of this linker that contains the IZI groups, the for subsequent conjugation of different linker payload could be realized simply through click chemistry reaction with the IZI. And as you can see, the resulted DAR is highly uniform at DAR2 and with pretty high SEC purity and can be easily scaled up at in the lab scale process. And additionally, we have developed a single hydrophobic chromatography step to efficiently remove the excessive enzymes, MTGs, and excessive unreacted linkers through a one-step chromatography step purification. And the second key trend is increased utilization of antibody fragment in the conjugation applications. So we have worked with clients in different AXC fields, such as the RDC, the AOC antibody nucleotide conjugates, and quite a number of cases for in vivo CAR-T applications. And given the very small size of the antibody fragment, as well as their simple structure, there are a number of key advantages of using antibody fragments in these applications. For example, their superior tissue penetration and delivery. It's very easable and flexible in engineering and allows conjugation. For example, you can easily realize site-specific conjugation by introducing cysteines at the C-terminal of these antibody fragments to realize specific conjugation. And also improved safety and drug-like properties. And there have been a number of different types of fragments, such as utilizing these applications, such as Fab, single-chain FV, VHH or nanobody or Fab2 fragment. So it's worth mentioning that ProBio has also established our CMC platform process for these various different type of antibody fragments for VHH, scFv, and Fab. So for the cell line development, which only takes roughly 10 weeks from DNA synthesis to top 6 clone production, and the titer of expression could be up to 15 grams per liter. And we have established our dedicated downstream purification and analytical platforms for these different antibody fragments to help accelerate the speed of and quality of the overall program. And we adopt light CMC strategies for these antibody conjugates programs, and particularly for nanobody like VHH. We can take as fast as 3.5 months from DNA synthesis to IIT material production, and as fast as 4 months from DNA synthesis to tox batch production for IND applications. And we offer flexible GMP production scale from less than 50 liters up to 2,000 liter scale. And I think there should be 11, but anyway, this so far over the past few years, we have generated over 11 antibody fragment CMC projects, and with the most advanced VHH projects has received IND applications from the US, Australia, and China. With that, I would like to thank everybody for your time, and happy to take any questions you may have. Thank you.